So thank you. Good evening. Let me just say a word to welcome Professor Michael Snyder, and then we will start the lecture. Ladies and gentlemen, it's my great honor to welcome Professor Michael Snyder among us today. Saying that he is one of the most influential scientists of our time would certainly be true, but probably unnecessary. Everyone here knows that he is a person who is reshaping the very paradigm of modern medicine. Professor Snyder is the Stanford W. Asherman Professor of Genetics and the Director of the Stanford Center for Genomics and Personalized Medicine. His groundbreaking work integrates genomics, proteomics, metabolomics, the microbiome, and digital health data, showing us that medicine doesn't have to focus solely on disease, but can instead help us understand ourselves and stay healthy for as long as possible. To meet Professor Snyder, to welcome him into our community, and to have his ideas shape the way we think about medicine and health, that's truly a dream come true. So I won't take any more time for making the dream last a little longer for all of us. Professor Snyder, thank you that you are here, and welcome.
Please, microphone on.
Oh, yeah. Let's see if that, how about now? All right, working? No? Yeah, there it goes. All right, great. Wow, it's picking up. Okay, well, it really has been a pleasure, as I say, to be here. I'm just having an incredible visit. I'm going to talk about our work trying to transform healthcare and the way it's done, but I was asked to give a few words about my background. So I grew up in rural Pennsylvania, just outside Philadelphia, which there were more cows than people by a lot. And it was very humble. We didn't have very much money. I did go to college in nearby by, I guess, certain standards, but far away in my case, because people really mostly stayed in this rural area. I went to New York for my undergraduate, where I became a chemistry major. And at the very end, I took some biology, and I thought, well, this is a lot more interesting. So I went to Caltech for my PhD, I hope you can say okay, where I actually got a PhD in biology, although I worked for a chemist. Recombinant DNA was brand new back then, and people were just cloning genes. So I am dating myself, and that's what I did as well. And then I moved for my postdoc to Stanford, actually, where I worked on yeast genetics and such for quite a bit and developed several technologies. And then from there, I actually went to Yale for my first faculty position, where I continued working on yeast, and that's when people used to study genes and proteins one at a time. Some of you may remember those days. And some of you still do that, and there's nothing wrong, but I guess what we did was we tried to look at studying the whole system at once. We were doing cell biological problems like chromosome segregation and cell polarity, and so that's when we started jumping in and tagging and studying all the yeast genes, all 6,000 at once, and then actually applying a lot of that to humans. And then 16 years ago, I moved back to Stanford when they were looking for a new chair. I was very happy at Yale, but Stanford has certain unique advantages, and I wound up going back to there and basically taking some of these systems concepts and applying it to medicine, and that brought me to where I am today. So I don't think anyone would have guessed that this guy growing up on this farm area in rural Pennsylvania would be chair of genetics, which I was. I just stepped down a year ago, but that life has strange journeys, and that's how I wound up where I am. So now I'm going to jump into science, which I enjoy talking about much more than myself, so that's where we're going to go now. So I can tell you what motivates us, which is that We think the healthcare system is broken, as you've heard already. And in fact, in the US, certainly, we practice sick care, not healthcare. People rarely go to the physician's office while they're healthy. And the consequence is that we tend to treat people rather than, again, keep them healthy. So there's a delta, if you will, a discrepancy between people's health span, the amount of time they're healthy, and their lifespan, how long they live. And it depends how you quantify that, but that delta is 11 to 15 years in the US and certainly most parts of Europe, meaning people are living the last decade of their life unhealthy. And we think it should be people live long, healthy lives and then just, pass away. So, but better than hanging on in a sick condition, I would say. Probably at the same time as you do extend health span, you would probably extend lifespan as well. That would be hopeful as well. I do believe we could engineer people to live forever, but that's a very different lecture than the one I'm going to tell you. We really work on health span. Now, even the way we practice healthcare, if you think about today, it's actually very antiquated. Typically, what you'll do, at least in the US, you'll get in a car, always at an inconvenient time to travel to a physician's office, where it hasn't changed a whole lot. There are some new gadgets there, mind you, but it's not all like dentistry, which is way, way more advanced, where you can get same-day crowns. A lot more technology is integrated into dentistry than it is into medicine. While you're there, they will stick a needle in you, take lots of blood, and from all that blood and time you're there, they actually don't make that many measurements, some of which are of dubious value, like resting heart rate in a doctor's office can be all over the map, depending how nervous you are. So again, that may not be the best way to measure people. And then from those measurements, they'll treat you based on population averages. We want to change all these steps, and that's some of the themes you'll hear about today. It is a research study, but we do try to get this out to the broader community. I was going to say, just from this standpoint, treating people based on population averages, you've been told since you're little, your temperature, at least in the US, is in Fahrenheit, 98.6, although you can see what it is in centigrade. But the point, first of all, that number is wrong. It's more like 97.5 from this study, but there are others say, big one out of Stanford measures at 97.7. More importantly, though, there's a spread. This is the 25th quartile, that's the 75th quartile. And so what that means in today's world, if your normal healthy baseline is here, they measure you at 98.6, they'll tell you you're healthy, you're normal, everything's fine. What are you doing here? Go home. But I guarantee if you're up 4 degrees over your baseline, you're not healthy, something's off. And that's a big theme of our work. Know what your healthy baseline is so you can detect shifts from that, no matter how yours differs from the person sitting next to you. Now, I think we all know our health is influenced by many things. It's influenced by your DNA, but that only accounts for about 16% of your lifespan. So it's a small fraction. 84% is due to other factors, including lifestyle, such as activity, food you eat, stress, as well as environmental exposures. And we're in a world where it's now possible to quantify that. Some very exciting work is going on here on the environmental exposure and probably other areas as well. We work in some of the space too. And even if you don't quantify this, some of it's easy to quantify now, like activity. Some of it's still a little clunky. But we can, even if you don't quantify this directly, you can quantify its effects by doing deep measurements on people. And that's what we set up when I moved to Stanford 16 years ago. This idea of doing what we call personal omics profiling, where we'll collect blood, urine, and stool, and actually other samples. And we'll sequence the people's DNA once. And then from the blood and urine, we'll make very, very deep measurements. This is DNA methylome, measure their RNA, and proteins. These will be out of peripheral blood monocyte cells, so immune cells. Cytokines we'll measure, and proteins we also measure out of plasmid. Same with metabolites and lipids, and some of that we'll measure out of urine as well. We do study the stool microbiome. I won't get time to talk about that much today, as well as other microbiomes. This is on top of deep clinical testing, questionnaires like stress surveys, things like that. And then we brought in wearables when they first came out as fitness trackers, thinking they might be powerful health monitors as well. And then we do have some advanced tests like stress echocardiograms and a lot in the area of glucose control, which I'll spend some time on today for a couple of reasons. One is I'm a type 2 diabetic myself, and the other is that diabetes is a growing problem all around the world, and we think it needs new approaches. So one aspect out of all this is take very, very deep data on people, and the other aspect is to do it over time. And I should point out it's the smallest group of people that we've been studying now for over 12 and a half years. We'll sample them every 3 months while they're healthy. And if an adverse event comes along, like a viral infection, we'll typically take 5 to 7 more samplings. So you might say, why are we doing all this? We're trying to understand what a healthy profile looks like, not a sick profile. How's that change over time? What happens when people first get ill? What's the difference between you and the person sitting next to you? It turns out that's more different than a healthy you and a disease you, which I won't get into too much today. And a big part of this is, can we use advanced technologies like genome sequencing, wearables, things like that, to better manage people's health? And I can tell you when we started this at the time, physicians did not like us sequencing healthy people's genomes. They said, look, you're going to turn everybody into hypochondriacs, and it's going to cost millions of dollars. This is a bad idea. And of course, we did it anyway because we think it's a good thing to do. Most have warmed up to it now, not all. Most physicians still don't like the idea of wearables because they think it's a lot of work and it gives a lot of false positives. Again, we'll talk about that in a bit. And then we'll also I'll mention whole body MRIs. I guarantee physicians don't like that. So we'll get to that later too. Anyway, of course, we did all this profiling. And I can tell you just from the first 3.5 years of doing this, we actually made, from the 109 people, 49 major health discoveries. So nearly half the people learned something about their health, and it spanned all these different areas: hem-oncology, cardiovascular disease, metabolic disease, and so on. And some were a pretty big deal. I should say these are all found pre-symptomatically, no symptoms. But again, some were important, like we caught someone with early lymphoma. These 2 are pre-cancers that can convert to aggressive cancer, so it's nice to know about them. 2 people had serious heart issues, one picked up by genome sequencing, another by wearables. And that was another lesson. No one technology found this. Sometimes it was a genome sequencing, sometimes the wearables, sometimes it's the omics. And the way we like to think about this is that if your health is a 1,000-piece jigsaw puzzle, we're trying to get 600 or 700 pieces versus 5 or 6, we would say today. We get very, very poor pictures of people's health with the way we do things today. And so again, these could all be uncovered pre-symptomatically. Now, because here I am, the father of genetics, I will say that genetics turned out to be very, very important. And in fact, these are Mendelian diseases. How many of you have had your genome or exome sequenced? Some, and they're all in the front. What does that tell you? Okay. Anyway, I do think when, 10 years from now, most of you will probably raise your hand because there is some value to this. So from the first 70 people, 12 actually learned something pretty important about their genome. So these are what called single gene mutations, Mendelian mutations. And a classic is BRCA, right? You've all heard of BRCA. If you have a BRCA mutation, it puts women at high risk for breast and ovarian cancer. One individual who was an orphan actually discovered this, so they'll get monitored more. 3 people had serious health issues uncovered by this. So this person, actually, a pretty young fellow, had a mutation in this gene and then did a follow-up, and sure enough, he's got a heart defect. His father, in fact, died of a heart attack. And so we think this is the culprit, the cause of the mutation. 2 people had a mutation in this gene, which puts them at high risk for certain cancers. One did a whole body MRI follow-up and had actually early thyroid cancer, also a pretty young person, had that removed, so they didn't need thyroid replacement therapy, so that was important. 9 individuals were thought to be type 2 diabetic coming into the study. One of them, once we sequenced, we realized they're actually a MODY mutation, and at the time, that was treated very, very differently from the way typical type 2s were treated, so they switched their medication, went on a better form, and that was very valuable. So genome sequencing, even though it's minority, actually still, for many people, this is potentially life-saving, pretty valuable. Now, most diseases are not Mendelian diseases. They're complex diseases. You may know this. And so single gene mutations, again, are things like BRCA. Here's another case for the MODY gene I mentioned. But for these complex diseases, these are generally thought to be due to common changes, common variants, if you will, a very small effect. And the way people deal with this now, and it's thought that if you're at high risk, you've gotten a lot of the bad alleles, if you will, the bad variants. And if you're at low risk, you may have inherited some of the good ones. And so the idea is that the Mendelian genes are like getting clobbered by a sledgehammer, where this is more like death by 1,000 cuts. Lots of little changes that harm you. And so the way people have dealt with this now is polygenic risk scores. Have you all heard of polygenic risk scores? Yes? Okay, quite a few. Good. So the way this is done now is that people will sum over, they do these GWAS studies. I see my slide disappeared, but you find loci that are genetically associated with disease. It's done in a simple association. It doesn't work that well. But these days, what you'll do is you'll sum over thousands, if not millions of variants. These are common diseases. There are many more, of course. And you build these polygenic risk scores. And it works okay for the top 2 to 4% of people. If you look at something called odd score, it works pretty well, but it doesn't work for most people. And we think there's several reasons for this. One is they ignore the single gene variants that can contribute to this. And they also, if you've ever done genetics, it's not linear. Meaning if you cross these things, they rarely, it does for Mendelian cases, but not for other situations. Genetics is often nonlinear, meaning 1 + 1 is not 2. The effect sizes can be much larger. And this is where the new AI methods turn out to be very, very powerful. So we got involved in this quite a few years ago before AI became so popular. And basically with these new methods, they're nonlinear, so you can pull together. And I'll summarize one part of this, but we started by doing 2 different approaches, one looking at abdominal aortic aneurysm, where 3 genes were known at the time. It's known to be highly heritable, 70%. Well, with our new AI method, went to 60 genes. You can look that up. I assume we can share these. I want to tell you about some of our work with ALS and COVID. Here we did use GWAS signals, these association signals, to actually find new genes. And they wind up finding these new AI methods anywhere from 10 to 100 times more genes than existing methods. So I'm going to walk you through the ALS one first and then the COVID one in a minute. And it will get a little involved. So if you're more medical, your eyes may glaze over. But just hang in there. We'll get back to more health monitoring in a second. So the idea here is the following, that you can... Most GWAS signals, these genome-wide associations, they land in regulatory regions of genes. And what you can do is you can map these out. So ALS, you may know, it's about 61% heritability. So it is fairly genetic, but the underlying basis is not very well known. There's a very large study, this one here, that mapped many cases and many controls, and they wound up finding about 7 genes initially, then later 15 associated with ALS. And what we did was we went out and mapped out the open chromatin regions of motor neurons, which is what ALS is thought to primarily affect. And you map these out with some of these new methods, ATAC-seq and ChIP-seq and things like that. And you can map, there's 100,000 regions within the genome. It winds up being about 5% of the genome is regulatory. And then what you can do for each region is computation intensive. Say, give me an enrichment for GWAS signal, not just the significant ones, all the GWAS signals for every one of those 100,000 regions. Is there a mixture that shows enrichment in cases versus controls? And that actually works well. We went from the 7 known genes to 690 genes. And for these complex diseases in general, people don't identify much of the heritability of the genetic component. And there was only 6% known then. We brought it up to 36%. And we did find many of the known genes. For those of you who follow ALS, some of these are very famous. And then we didn't get them all, but we got a lot. The others fell just below the threshold. We use a conservative cutoff. And then we found lots of other genes implicated in ALS by misexpressions, things like that. We went on to show the genes. First, we validated another cohort, and we show the genes that we identified, these 690 are lower in expression in ALS patients, their brain tissues as well as iPSC-derived motor neurons. And we could even show the genes tended to function in distal regions, so we could actually map out some biology. And we took this one step further, where there was one gene at the top of our list that looked like it might have Mendelian forms of it. So we went in and recreated that mutation in iPS cells, derived motor neurons, and bottom line is we could show they had ALS kinds of defects in these motor neurons, including mislocalization of this gene, which many people think is responsible for ALS. I don't know how many of you followed this. Point out of all this is we could go from this new method to finding genes that we think are pretty powerful, and now we're actually trying to integrate this information into the clinics, that we think this could be quite powerful. And so the bottom line is that this approach does work well. What happens if you don't know what the responsible cell type is? So then you can do this different approach with single cell data, which you can combine. So COVID severity actually has a genetic component you may or may not realize. So people over 65, their immune system declines, and that's why they can get in trouble when they get COVID. But for people who are under 60, many of them actually get severe COVID and die or wind up in a hospital as well, and that's thought to have a genetic component. Anytime kids or younger folks have a problem, it's thought there might be some genetics involved. Now, for this case, we didn't know where the signal might sit, what cell type. There are 19 cell types in the lung. We didn't know which might be the one responsible for severe COVID. But these days you can do single cell multiome, which does both this attack for mapping open chromatin regions as well as RNA. Again, this is getting a little heavy duty, but you can do this for all 19 cell types in this new method called multiome analysis. So we mapped out the open chromatin regions for all 19 cell types. Then one by one through each of the cell types, you could do the same thing. You could look for enrichment of signals relative to controls. And the bottom line is we could find over 6,000 regions mapping to about 1,400 genes and across the different cell types. Now, it wasn't so clear which cell type, again, was responsible for COVID. Some people said, well, it's the epithelial cells in the lung. Others said it's the immune cells, either a natural killer or T cells. But with genetic methods, you can actually show most of the signal sits in these natural killer and T cells. So we think this is the underlying basis for the genetic cause of severe COVID, again, in younger folks. And we could go on and show actually these genes are lower expressed in certain cells, especially the natural killer cells. We even can map the subtype of natural killer cells. And what was amazing was that for the most severe cases, we could explain 77% of the heritability with this gene list. So the point out of all this is that with these new AI methods, you can really analyze the genetics at a level that's never been possible before. And we have newer methods now that I won't tell you about, but they actually are powerful for finding things you think are well studied, like diabetes. But most complex diseases, they just don't have the heritability well mapped out. And these approaches are really getting us a lot more information. So that's what I was going to tell you about genetics for now. Other things we could learn from this study, remember we're profiling people over time. And so we can actually look at how they're changing over time, which we interpret as aging when we're looking at healthy time points. And so by looking at the biochemical pathways that change, again, we can see what's going on. And it turns out we interpret this as aging, and everybody, it turns out, is different. So this person, their top biochemical pathway that changes is their cardiac hypertrophic signaling pathway. They're a cardioager. This person's pretty typical ager. Their immune system changes, it turns out. Their metabolic system changes. That's what normally happens. We think with as few as 4 or 5 measurements, we can see exactly how people are shifting over time. We call these aging patterns ageotypes. And at the time, we had 4: kidney, liver, metabolic, and immune. And so the people over here, each column's a different person. These 4 people over here, they're aging in all 4 categories. This one's not aging much for kidney, but for the other 3, this one's a kidney ager, kidney and metabolic, and so on. So we can see how people are aging based on their biochemical changes, and it turns out there's clinically actionable information with these patterns. So for example, the metabolic agers, they have hemoglobin A1C, which is a marker of glucose, and other things shifting, and the folks who got this information, some of them shifted their lifestyle. They either lost weight or exercised, and they improved their metabolic profile. Now, I'm not saying they got younger, but they certainly improved their metabolic markers. I'm going to come back to this in a minute. Now, the other thing is because we're following people over time and the folks are at different age categories, we could see how they're changing. And most people assume aging is linear. Well, you just gradually get older and then it's known later life things will happen and you pass away. But we could actually look at this and say, well, maybe aging isn't linear at all. Maybe different pathways are changing more at certain times than others, like 30s or 50s, what's going on? And are there certain times where there would be the most changes occurring? So we had 135,000 measurements when you add everything up. And sure enough, it turns out there are 2 times most processes are not aging linearly. And there are 2 times when there are the most changes. One in the 60s that we expected, and another in the 40s. We said 44. Don't take that number too seriously, but you get the idea. It's in the early to mid 40s. And so for this one, we can ask for both, we can ask what pathways are changing. And this was expected. We saw skin and muscle mass changes. It's well known people get sarcopenia as they get older. Their immune system declines. That's why you get vaccinated. We also picked up other things, carbohydrate, kidney function, especially oxidative stress showed shifts as people hit that period. And again, we expected that, but did learn new details. This one was not necessarily expected, but anecdotally, a lot of people have run into this, and some of you may be nodding your heads because you've run into this as well as you hit your 40s. But there we also did see as well skin and muscle changes. We did see shifts in lipid metabolism, which I think is significant because I think people's fat distribution does shift as they hit their 40s. The way their body build is different. It's different as they hit their 40s compared to their 20s. And we also saw shifts in alcohol metabolism and caffeine metabolism, which was true for me personally. I had to stop drinking coffee after noon or it'd keep me up all night when I hit my 40s. So anyway, the point out of all this is that you do these, and these, once again, it's actionable information. You can use this to look at these things. So the point out of all this is we're not only we have personal aging patterns, but there are certain things you can look at in bulk, like lipids and stuff, where you might keep an eye out for that as you hit your 40s, and if it goes off, maybe take statins. Now, one question I get asked a lot, this is a great research project. How are you going to scale this? How are you going to bring it into medicine? Well, I'm in Silicon Valley, so we form a company. That's how we think the easiest way to get this out there is. So we formed this company called QBio, and they do a medical version of what I just said. They won't do microbiome just yet because we don't know how to act on that, although that's starting to change. They will do many other things, and they've also added whole body MRI. Now, as I mentioned earlier, if you ask any physician today, should you do whole body MRI, pretty near 100% will say absolutely not. And do you know why? What's that? Money, oh yeah, sorry. You'll notice in a minute I wear hearing aids. Yeah, so money is certainly one. There's another reason why physicians don't like it. Yeah, you see things that like nodules in people 100% of the time. If you go high enough resolution, women have them in their ovaries, nodules, and men in the prostate. If you go high enough resolution, other sites as well. If you ask me, should you do whole body MRIs 100% of the time, I'll say absolutely you should. Because the issue isn't do you have nodules, the issue is do you have any growing nodules. And the way you know that's from your longitudinal profiles. And I've done, now 21 whole body MRIs in the last 9 years. I have 9 nodules. I know exactly where they are. I'm happy to say none are growing. But I can tell you for a friend of mine this happened. He actually woke up one morning, couldn't move his arm. They took him to emergency, looked at, they did a scan on his back, basically. They saw a big tumor on his spine, and they cut it out. And then he recovered and got his mobility back, but then a month later did a whole follow-up on him, whole body MRI then, and they saw 3 more nodules on his spine. So the question is, was it there to begin with, or had it metastasized? There's no way to know because they didn't have a baseline. So that's the power of this. You really do need to know your baseline. I'm a believer. And we need to get whole body MRIs dirt cheap, by the way, so they aren't so expensive. Anyway, so you can see I'm a believer in data, as you can tell. So the other area that we work very hard on is this idea about doing remote monitoring and so-called Amazonizing healthcare. And we have 2 approaches, one with wearables and the other is home microsampling that I'll get into in a bit. So wearables are very powerful. How many of you are wearing smartwatches or rings or things like that? Yeah, quite a few. And more in the back, I'm glad to see. Great. All right. So anyway, you may or may not know I wear my 4 watches. my ring, my exposometer, which I won't have time to talk about today. But on top of that, I even have hearing aids, which I do use for hearing. You can see I still have trouble here, but they're also sensors as well. And the reason all these things are powerful is they follow you 24/7 as long as you keep them charged. They'll monitor you all the time. And some of these are pretty valuable measure. We would argue heart rate variability, resting heart rate, which all devices measure, and they're pretty accurate for those too. They're not as accurate for some of the other things, but that information we would argue is pretty valuable. And then they measure blood oxygen, galvanic stress response, conductance on your skin turns out to be useful, we think, as well, and sleep and other things. So we think these are pretty powerful monitors. And so back when they came out as fitness trackers, we put them on our cohort, because we thought they might be useful health monitors. But right away we discovered you can tell when somebody's getting ill, in this case from Lyme disease. This is actually me. I first realized I got Lyme disease from a smartwatch and a pulse ox. Now you can get blood oxygen right on your watch. You couldn't back then. It was 2015 when I was flying to Norway, actually, and I noticed that my blood oxygen was abnormally low on the flight. My heart rate was up to this low resolution, and it didn't come back neither came back to normal when the plane landed. And I did get a fever off and on. I visited a physician in Norway. I warned him it might be Lyme because 2 weeks earlier I had been in rural Massachusetts where the most ticks are Lyme infested, and I don't know how much there is Lyme in Europe, you may or may not know, and it's growing, right, because with climate change it's getting warmer. So anyway, and this is spreading more. But point out of all this is that I first noticed I was ill pre-symptomatically from a smartwatch and a pulse ox. By the way, he wanted to give me, I went to a physician here, he wanted to give me penicillin, and I said, no, I need doxycycline. Got a little tense there because most physicians don't like their patient telling them what to do. And he gave in and gave me doxycycline. When I got back, I tested, sure enough, I was positive. It's a very well-controlled experiment because I'd given sample before I'd gone, and I was negative, so I seroconverted during that time. So the point out of all this, again, pre-symptomatically, we could tell I was getting ill. I won't show you the data for this, but we could show what was true for respiratory viral infections as well, because I had 2 years of smartwatch data. We went back and looked at it, and every single time I was ill, as measured by CRP or not feeling well, I could see a jump up a resting heart rate before symptoms, 100% of the time, even one asymptomatic case. So then, of course, along came COVID, you may know, hit us in spring of 2020. This is an old slide, but various peaks. I think we're just going through a pretty big one in the US right now. If you think about how do we measure when people are getting ill, well, temperature, which is a 300-year-old concept, and it doesn't work very well for COVID, right? Because half the people don't get symptoms. I didn't when I got COVID. And so you can get better tests with antigen or PCR, but you're not going to do that all the time because it's not convenient, it costs money. So we think smartwatches might be a powerful way to go. So right away when this happened, we partnered with Fitbit. We had 53 people sign up very quickly, and we had 32 of them. Actually, sorry, this is slipping a little bit. That probably sounds better too. Anyway, 32 of them had COVID while they were wearing the smartwatch, and they had a diagnosis day and a symptom day. And this is our very first case. This is resting heart rate. So this person got symptoms here in red, diagnosed the next day, but you can see their resting heart rate jumped up 9.5 days prior to symptom onset. Okay, so very clear signal. And it turns out for 80% of those 32, so 26, we could actually see a jump up in signal at or before symptom onset. Again, in 80% of the cases, the median was 4 days. So we wrote a real-time detection system where we follow people in a circadian fashion, look for a jump up in resting heart rate. And if it's up for too high or too long, it gives a red alert. You can all sign up in our study. Here's how you join, because we want to perfect these algorithms. And that also works. So this is one of our first cases here, same number, 80% of the time. And here is an example here. This person got symptoms here. They were diagnosed the next day, but they're getting red alerts for 3 days prior to symptom onset. So we think you need as little as 2 beats per minute. It's pretty easy to pick up this situation. And in fact, it's so sensitive, it's more sensitive then an antigen test will turn it, but it also picks up asymptomatic cases. It works for Apple, it works for Fitbit, it should work for everything. And the bottom line is that this is an asymptomatic case with Fitbit. This person was diagnosed here, but they were getting red alerts for 2 weeks prior to symptom onset. This is my case. We have others like this to show that actually it's more sensitive than an antigen test. I woke up one morning in spring, this is a real date, feeling a little congested. And I wasn't sure, well, was it allergies? Am I coming down? I'm not sure. So I did an antigen test, and that came out negative. I looked at my smartwatch, it was positive. Now, I was due to fly to New York City, so what did I do? I went on the plane. Big mistake. Before I could go to the meeting back then, you had to get tested. It was, pretty early in the pandemic, relatively. And so I tested positive the next morning. So I had to spend the whole week in a tiny little hotel room in New York City, all because I listened to this and not that. So it's more sensitive, but it's not specific. So we think this is important. So in fact, we asked for the non-COVID people, what other things cause red alerts? Well, intense exercise, if you run a marathon, if you drink way too much, you'll not one glass of wine for dinner or even two, but if you really tie one on, your heart rate will be up and you'll set off red alerts. It turns out workplace stress is the #1 trigger of red alerts. I know there's none of that going on here, so you probably don't have to worry about it. The point out of all this is that it's also a mental health detector as well as, and we're actually trying to turn that into a mental health assay to go along with this. Now, we think this is the tip of the iceberg. If you think about smartwatches, right, they're shining light into your blood, and they can measure hemoglobin, various things, and quite a few parameters. And so this is with a 2016 smartwatch. We actually looked for association, once again with machine learning and AI. We looked for associations between clinical measures like red blood cell count, hemoglobin, and features of a smartwatch. And I won't get all the details, but you can basically, again, those you know how to do this probably know what I'm talking about. You can take these features and make these predictions, and it's actually decent. It's not good enough for a clinical grade measurement, but it's plenty good to see a shift from your baseline. So we think we can tell things like, this is relevant for anemia, this is relevant for diabetes and other things. You can capture all this. The other area that we push very, very hard on is this idea of continuous glucose monitors. Now, these are being used for insulin-dependent type 1 and type 2 diabetics. Just curious, how many of you worn one of these? Yeah, not as many. Well, they actually were over the counter here in in Europe before in the US, believe it or not. Our claim to fame is we started putting these on so-called normal and pre-diabetics. And what we discovered is that a lot of normal people, they measure your glucose every 5 minutes. A little background, 11.6% of people in the US are diabetic, mostly type 2s, and 33% are pre-diabetic, and most of those will become diabetic. And those numbers are getting worse. We think the diabetes endemic is worse than the COVID pandemic, actually. So we put these on normal people, as I say. Some have pretty good glucose control, not much problem. Some are moderate spikers, and some are severe, just as bad as diabetics. I think it's missed because of the way they were diagnosed, you'll see in a minute. And so informatically, we classify people into what we call glucotypes, spiking patterns. And then we, as an Aaron Siegel, discovered right away that different people spike to different foods. By the way, I bet a lot of you are pre-diabetic and 90% of pre-diabetics don't know it. So there's a good chance a lot of you are pre-diabetic and don't know it, and we think you should. So anyway, this is from Aaron's work. Some people spike to pasta, some to potatoes, some to white bread, some to brown bread. This is from Aaron's work. He showed there's an individual that spikes to banana but not a cookie. Probably the only person on the planet doesn't spike to a cookie. Most people do. This person spikes to a cookie but not a banana. That's more normal. Okay. A recent study we just published, we had 55 people eat 7 different carbohydrates, identical amounts. I think it's 50 grams. But the carbs are in different forms, beans, berries, you can see different things, potatoes, rice. Most people are like this. They did it at least twice, sometimes 3 times. Most people are like this. They're rice spikers. And believe it or not, for many people, possibly most, rice is worse than ice cream. Actually, very high glycemic. So you can see, so most people are like this. Interestingly, Asians are spike more than non-Asians even. But there are people like this. These are bread spiker, grapes, pasta, potatoes is yellow. So again, different people spiking different carbs. What's going on? We think this is due to the subtype of diabetes. We tend to lump things in type 1, type 2, the Modi thing I mentioned before. But we actually think type 2 has many different subtypes. It's not just 2. And if you think about it, it involves many organ systems. There's liver, pancreas, brain, muscle. These all influence glucose control. And many signaling pathways. Insulin's famous. But so are these GLP incretin pathways you may have heard about. They're a big deal now. Glucagon. All these actually influence your glucose levels. And so we think that it's probably heterogeneous systems that are off, and that's what's going on. So we went in and took, in this case, I think it's 36, we've done many more now. These are typed by their level of glucose through hemoglobin A1C. Blue means they're so-called normal. Yellow means they're pre-diabetic, and red, a diabetic snuck into the study too. Yeah, we're shooting for just pre-diabetics, but anyway. You can see everybody's different. There's muscle insulin resistant, beta cell defect. They don't release insulin from the pancreas. Incretin are these new GLPs, and hepatic insulin resistant. You'll see, here's a good example here. I think I have yeah. This individual is very muscle insulin resistant, intermediate for beta cell, normal for incretin, intermediate for hepatic. This person, very strong incretin defect, sensitive for muscle, and so on. So people are different forms of their glucose dysregulation. And we actually, these days, can tell, at least for two of these, muscle insulin resistant beta cell, we can actually tell just from the shape of a curve, if you've ever done these. People have very different shapes of their curve. We think that reflects the underlying defect in the glucose regulation. And we can actually build predictive models. So now, with one of these glucose monitors you can buy in a store, you can actually drink a shot of glucose, and for insulin resistance, muscle insulin resistant, beta cell defect, we're quite predictive of whether you have a problem there or not. Not so much for the other two, we're still working on these two. But that test normally costs thousands of dollars. We think you'd be able to do it for about 50 bucks in the future, 50 US dollars. So anyway, that's a little side note. All right, so what? Why do I care about my diabetes subphenotype? Well, it turns out the diabetes subphenotype determines what foods you will spike to. So if you're muscle insulin resistant, you'll be a potato spiker. That's shown here in yellow. You'll also be a pasta spiker, but not if you're sensitive. Okay, so you can see these are much lower here. I won't show you the data, but for the beta cell defect, the result's the same for potatoes. You'll spike the potatoes, but not other things. So you can start matching the foods based on your subphenotype to the foods you should or shouldn't eat. We could take this one step further. We can basically, from people who are subphenotyped, we can track their glucose along with their activity, and they do food logging. And the goal of this is we track them for several weeks. The goal of this is to try and match people's, if you will, lifestyle and things they do with their glucose levels and such. And it's not just what they do, but when they do it. So timing is important. And so we take all these and then we build machine learning models to be able to predict glucose levels and actually their subphenotype. So as an example of this, if you eat this is, if you will, glucose level by hemoglobin A1C. If you eat your first big meal in the morning, you'll have lower glucose, but not if you eat it late at night. That's here. Starchy vegetables, not so good. You'll have higher glucose, but fruity ones are good for you, lower. Most people don't sleep enough, so if you don't sleep enough, your glucose will be higher, and so on and so forth. So we can actually, some of these were known already, but we find lots of new things. But we can also do this for predicting subphenotypes. So, and here's a personal example. If you exercise more, you'll lower your muscle insulin resistance. The reason this is a big deal, I can tell you personally, I actually I have a beta cell defect. That's my subphenotype. So I actually used to be a runner. I shifted to lifting weights. You can't tell, but I gained 10 pounds of muscle mass. And I did it to try and control my glucose. And it failed miserably. And that's because increasing my muscle mass isn't going to help my beta cell defect, right? I can gain 100 pounds of muscle mass. I'm still going to have a beta cell defect. It's not going to help my glucose control. So we think this is important. So if you know this, well, then actually building muscle mass is helpful. So we've been, anyway, we're pushing this concept even further. And we think it also affects the drug. So I actually respond well to a drug called rapinolide that actually promotes release of insulin from the pancreas. I'm a metformin non-responder, actually, which is uncommon, but that is true. Yeah, anyway, so you can see you match your drugs to your subphenotypes as well. So no, we don't think it's useful to think about diabetes anymore as a single, or type 1, type 2. It really should be broken into many subtypes. I'll tell you one more round of stories here. I think I have time. Yeah, well, then I'll wrap up, which is this whole microsampling, the idea of giving little drops of blood so that you can measure many more things in a convenient fashion. And so we started with these Mitra, these devices. We spent 7 years perfecting this. You give little drops of blood. For this one on your fingertip, there's another in a minute. I'll show you that, again, is on your shoulder. You overnight it to the lab, and then now we measure about 7,000 analytes on this, okay? And by doing metabolomics, proteomics, lipidomics, some of these are stable, some not. We know which ones are and which ones aren't, so we can correct accordingly. Then we did some really fun studies. We had 32 people drink the shake. I don't know if they sell it over here. They were fasted at the time, then we profiled them afterwards. And what we discovered, they're not surprised, hundreds of molecules change, and we can see, you know, some pretty important molecules change with targeted assays like insulin, C-peptide, things like that. That's one of the incretins, even inflammatory markers. The cool result from this was everybody reacted to the shake differently. So I'm just showing 3 carbohydrates here. These are different people. This person, their carbohydrates goes way down when they drink the shake. For these people, it goes up, up, up. This one's flat, that one's down, that one's down. Informatically, we can group people into 5 groups. So for this group of people, these are classes of molecules, if you will. Brown is inflammatory. So for this group of people, gray is the average of the 32. For this group of people, their inflammatory markers goes down when they drink the shake. For this group of people, it goes up. Same with that one. Same shake, pro-inflammatory on some, anti on others. Very personalized response to food. We can take this one step further. We can actually have, there's a person who gave basically a sample of blood every hour for 7 straight days during waking hours while wearing a smartwatch, a CGM, food logging, etc. And the idea, once again, is to correlate physiology and activity with biochemical and other changes. And you find hundreds of associations, or thousands, I should say, between heart rate, glucose, steps, and you do these time-lag correlations. And I'll just show you 2 examples. This is an obvious one. Your insulin C-peptide goes up after glucose. It should, and it does. But we can measure it precisely in this person. Here's one that wasn't known. Alpha-synuclein shows an interesting pattern. We think it correlates with stress. So if you're at risk for Parkinson's or dementia, you might want to know this with the idea of trying to reduce activities that would elevate that. So you could try to, again, diminish that and perhaps push off getting dementia or Parkinson's. That's the idea. So we're testing that now. I'll just say we can bring in our wearable and other omics data into dashboards so you can display them. And this is Mike Snyder's world. I don't think everyone will agree with this, but I'm a believer we should be getting genome sequenced before birth, or at least after. A little late for us, obviously, but not completely late. Anyway, and then with other deep biochemical and wearable data, you can build personalized AI monitoring systems to track people's health, and we actually have been doing that. And so once again, the way we try to get this out to Silicon Valley, and it's hard to incorporate these things into the healthcare system in the US, so you actually have to have people pay out of pocket. So anyway, we've spun off all these companies. I want to tell you about this one, IOLO, which does microsampling. It takes collects drops of blood from your shoulder, and basically they measure about 650 metabolites. And so, yeah, and it covers these 20 wellness categories, things like inflammation, oxidative stress, hormone health, et cetera. It even measures your biological age. And as an example, here's an individual whose biological age is older than their chronological age. But the nice thing is you can break it down into ageotypes. So their inflammation markers aren't so bad. They're actually pretty good. Their metabolic age, touch older, but their heart age is a lot older based on various markers. And so, and these are markers found in the scientific literature. So based on this information, they use AI to take your data and data from the literature and make personalized recommendations, not just exercise more, eat better, but really specific, you should eat this. Here's what you've been eating, you gotta eat this. And that we think will improve things. So bottom line is, I won't have time to go through this, but we think bringing in lots of data and doing remote monitoring, we think can really change the way healthcare is practiced. And I'll just wrap to say that this work is done by very talented people, postdocs and students over the years, and so these are the ones who have done this. Because I'm a little late, I won't have time, but they're the ones who deserve all the credit. And so, excuse me, I have time to take some questions, a few questions, I think, if you have any. Thanks again for having me here. How many of you are ready to go get a CGM now? Oh no, nobody raised their hand. My lecture was futile. Yeah. Okay.
Okay, thank you for a wonderful lecture. And I think and I expect so many questions, especially of coffee lovers and people who are interested in their health. So please, I'm opening the discussion. I have a microphone.
So thank you for a lovely talk. I'm a big fan of what you're doing. And you place it as healthcare is broken because we simply didn't have the methods, whereas I would argue that healthcare is mainly broken because the human nature is broken. And there are lots of studies which show that we simply don't care about threats in the future. And you can think global warming or the 40% who are obese and well know that it will kill them. So yeah, I would say like all this will certainly help maybe 10% of people who care about such things, but like the rest will simply not care. So do you think about this?
Yeah, we think about this a lot, and your point's very well taken. I think what it means is we need new approaches. For anyone who wears a CGM, and we've run studies on this, they will improve their time in range, it's called. There's a recommended range you keep your glucose, without even trying, just by wearing a CGM, because it's so visual that when you eat something, and a good example I'd like to use, I told some people this earlier, that a New Times reporter came to me, he ate salmon on salad every day for lunch. He thought he was having the healthiest lunch ever. And then he put on a CGM and discovered his glucose was spiking through the roof. And the reason, it turns out, was the sauce put on the salmon. So once he discovered that, he just left that off and he got the healthy lunch he wanted. So some things are very easy and it's so visual. And I can tell you, if you wear a CGM, you'll never eat the same again because you will see exactly what's spiking you and what doesn't. And if it does spike you, well, go do a 15-minute walk. And I didn't get into that, but that's some of the details that we've learned from our studies. So there's ways of mitigating this thing. And, and I heard someone nicely saying this at lunch today. They made sure they ate their salad before I think it was potatoes, if I recall right. So you get the idea that you can actually, so there's very simple things that I think people will do. But you're right, some of these areas are tough. I think you're well better positioned. I think there are two things that motivate. I'm probably rambling a little bit. Money and family motivate people. In the US, we don't have what you have. I hear your preventative health care is actually pretty good here. We actually don't have that in the US because people change their health plan every 18 months. So nobody puts a lot of money into you knowing 18 months later you'll be with somebody else. So we need to make this stuff dirt cheap, which we're trying to do. But then I think, again, we could incentivize people. I think you should get a discount on your insurance and such if you wear a smartwatch. I think you should get paid to get your genome sequenced because you're going to learn your risks and manage things better. So I think we could come up with creative ways to try to actually get people incentivized, because you're right, otherwise they may not be motivated to change. And you've hit the nail on the head, what's the biggest challenge.
Looking at your data from milkshake study and so on, and inflammatory data, I just wonder if you take into account allergies, people's
Allergies?
Yes.
Yeah, we know, I mean, some people do have allergies. I have allergies. The nice thing is you can track what's going on. You just look at their eosinophils and such. So we do follow that. Yeah, what seems to be healthy with no, and there's surveys that are supposed to check that kind of thing. On the exposed omics part, which I didn't present today, we can actually start correlating what's on the outside with what's on the inside. It's kind of fun, actually. So a good example is actually me. I get mild allergies. It used to be worse in the spring, and I always assumed it was pine pollen. And then when I started doing this tracking of exposomes and such, I discovered it correlated better with the eucalyptus. And I should have figured out ahead of time. All this stuff is, duh. I should have realized that earlier. And the reason is because I didn't really get them on the Northeast where they don't have eucalyptus. And I only got them in California and Florida, places like that. So, and they have pine everywhere. So I probably should have figured it out earlier. But anyway, so what does that mean? Well, it means probably I'm a little more careful in the spring around the eucalyptus tree, the giant one right behind my house. So. Yeah. Oh, and food allergies, yeah. Well, that's the thing. A lot of people don't know what's causing their food allergies. A lot of it's just instantly assigned to gluten. And sometimes that's right, and sometimes it's not. So I think we can measure now with the microsampling, yeah. So that's one of the next studies we're trying to run. Yeah, great question. And I don't know what the numbers are here. In the US, 10% of people have irritable bowel syndrome. And it's not unreasonable to think that that's either what they're breathing or most likely eating. Yeah. But we want to track all that. Yeah, great comments.
Dr. Snyder, thank you very much for your wonderful presentation. And you showed us a glimpse of how would be our medical future with your research and what you were showing us now. But I wonder, I would like to draw some words about how would be the role of the physicians in the nearest future, when this technology will be widespread. Will be the real role of the physicians and the medical patient, if patients can monitor themselves? Would you think the doctors will be disposable in a short of time?
Well, at some level, yes, but not completely. So I think, first of all, AI doctors will be superior for most things than other doctors because they can collect information. They get all the information in the world, right? And they can bring it to you. And I can show examples. I have 2 petabytes of data around me. And we can feed that into an AI program we have. And I have all these concierge doctors who all want to, measure me and look at me. So I get all this free advice. Some of them normally charge like $250,000 a year, and I get it for free, which is kind of cool. But anyway, with this AI, even all the information, all the expertise in the world, one of these AI programs told me 2 things none of my doctors told me. Now, one I'm not sure is 100% right, but it's not unreasonable, and it gives where the references where this comes from. And the other is actually quite correct about I should probably be taking zinc for a certain thing. So I think that will become common, but you'll need a physician, at least right now, for sure you need a physician to make sure things are accurate and such. But in the future, for common things, I mean, you guys probably Google up, that's what I do, if something goes wrong anyway right now. But imagine an AI version of that will be way, way better. So I think that for routine things, I view it to like airlines and tickets. I'm dating myself, but I remember before they had kiosks, right? And everybody's waiting in these long lines. And they were going to bring out these kiosks. Everybody said, nobody's going to use those kiosks, right? They always want to see a person. Well, of course, that's not true. Right away, everybody moved into kiosks. And you only see a person if you have a problem. And I think that's how medicine will be in the future. You just get 90% of it, the low-hanging stuff. You just grab from your AI doctor. We obviously need to get it working better than it is now, but it's not bad now. And right now, I would not want a doctor who wasn't using AI because it just draws in more knowledge than they can possibly have in their head. And I was telling Don about a new course we're running where you can actually make your own code and make your own thing. We're running a course that you can take and actually make your own programs for better managing your health, actually. So I think we're going to have a lot more power on the person. Now, whether most people do it, probably not, but maybe somebody in your family who's into this kind of stuff will do it for everybody. I don't know. Yeah. So they won't go away, but yeah, I think he might have been next, or maybe not.
Really, thank you for your inspiring lecture. And I'm doing my PhD research about the aging population, especially using the new technologies of the informal caregivers of the people with dementia. So this is my point because I'm a sociologist, not biologist. But how you raising an awareness of the using such kind of products and all of the new technologies, for example, for those target groups, how you're raising? Do you have a special educational path for them, or maybe you are organize the lectures? How you're introducing those AI tools and the other new technology tools for the people?
Well, I guess two comments there. One is I think we could be measuring people better, such that you could actually measure their interactions now with voice recognition or other things, and how much you're watching TV versus participating in social activities, and not only how much they're participating in social activities, but how much they themselves are engaged in this. And then you could come back with personalized recommendations saying, well, you're sitting at home on the couch too much, you should be getting out and socializing more. So you actually could build in behavioral incentives, and I think that's what we need. And I think to a point you alluded to, if you look at people live in the blue zone, so that these are areas in the world, and they're scattered about, where people live, excessively long, or unusually long compared to other places. They have four things in common. They're active, they eat well, usually not much ultra-processed foods. They actually have great social networks, either within family or in their community. And it's not so well cited, but I predict they sleep probably pretty well. So those four ingredients, and there are other things too, but those are like four really basics that everyone, and we could quantify all that and then try and get people to, deal with this. Now, people often say, well, wearing a ring makes me anxious, so I'm going to sleep worse by wearing a ring. But I think we have to figure out ways to, I guess, culturally get used to that. The analogy I like to say is, we're used to having, your car has tons of sensors, and you have a dashboard that takes that information, relays it back. You couldn't imagine driving your car without a dashboard. Yet we run our health without a dashboard, which is crazy. I think we need a health dashboard that's very active and very engaged and gives us feedback. Not to the point where it's harassing us. I turn my Apple Watch off. It harasses me too much. I hate it. But I do want the information stored in case I ever need to go back. So, yeah.
Hey, I'm just wondering, have you been collecting any gut microbiome data in your cohorts? And did you find any interesting patterns in that? Because there are several subtypes.
We have lots of patterns with the microbiome. We actually look at 4 microbiomes: skin, gut, nasal, and tongue. And there's all kinds of interesting things. We published a paper you could take a look at. And in general, the microbiome has been tough to act on. And I think it's because microbiomes are guilds of communities. So everybody's microbiome is very different. But the community itself has figured out a stable way to stay, again, in homeostasis with your immune system. You may or may not know you have as many immune cells in your gut, more in your gut than anywhere, 70% of your immune cells. So there's this balance going on. And we just haven't figured out how to make it for them. So there's some simple things. Have a diverse microbiome, that's good. Make sure you have enough bifidobacter and lactobacillus, those are good. But there's not a whole lot of other actionable information. Now, several companies will argue with what I just said, but I think it's just been harder. It is true that Akkermansia, if you take it, you can lower your hemoglobin A1C by 1%, which is pretty good. So there are some things that are there, but yeah, it's not, it will be, I think it's part of our research profiling. It's not yet incorporated into our other things that we've commercialized because it'd be hard to act on the information as it currently stands. Yeah.
Hello. Thanks for the nice lecture. I was wondering about those spikes, rice, potatoes, grapes, or whatsoever. Is it regionally correlated? Could it be like we are, for example, here, potato eaters, somewhere will be rice eaters, somewhere maybe, that's one question. Second, among those wearables, one of them device which is missing there and might be, it will be coming, it's a smart toilet. You know, for the metabolites, I've seen that the market is trying to get it.
Right.
But for some people, it will still be kind of awkward to be analyzed.
Yeah.
But as you say, it's important information. It's
No, I agree. I actually, believe it or not, proposed smart toilets back when we started this, and there are some out there now. They haven't quite got to robust. They're pushed pretty heavily in Japan, actually, you may know. And the one I've seen, the latest, is one that measures about 6 things, pH. The most important one's probably blood in your stool and things like that that could, you know, be an early sign of colon cancer or other cancers. So there are useful markers. We just haven't, you know, as much as we try to do everything, we haven't gotten. We're way behind on the mental health monitoring too. So that's what I would say. I agree. I think it's a good thing to have. I forgot what the first question was, though. Comment.
Sorry, the first, whether it's regionally.
Oh, yeah.
The spikes can be regionally or culturally connected.
Yeah, I don't know. You know, the fact that Asians spike more than non-Asians, even when they're same area, does say. So I think the spiking, we don't have everything measured. We are measuring the microbiome, but we don't have it built into our predictive models yet. So I think, first of all, where you live probably does have some influence on your microbiome and your immune response, all this stuff. So there's still many more things to bring. So I predict it has some influence. What fraction that is, I don't know, right? But that is the ultimate goal, to know exactly what's contributing these things so you can design diets and activities, again, to optimize health. And I didn't say, but that's actually what came out of that research study I did. And I forgot to say, but it should have been obvious, I'm 100% conflict in everything I've told you. And we have a CGM company that has built AI models, very predictive, quite nice, actually. Yeah. Well, I'm fine if we're going. If you're fine, I know it's warm in here, so I don't know if, feel free if you want to go, I won't be offended. Well, maybe just a little bit, but.
Yeah, thank you for a great talk. I was wondering about the potential of wearables if in the future integrating AI, for example, to making corrections as when relying to the ability of AI to track our day-to-day activities and then having more specific information from wearables. Could it be possible to replace many immunoassays, for example, or other assays? Thank you.
I think I got a lot of it, but what incorporating, do you want to, maybe I'll move closer.
The AI, for example.
Yeah,
AI
I think AI for sure is going to be part of everything with wearables.
Can wearables will be more specific then if, for example...
Will they be more specific?
Yeah, with doing for example, real-time corrections for our daily activities. As in the example, it was...
Yeah, I mean, they'll build models, so we'll all have an AI agent, I believe, that will be, and the wearables will be an important part of that. But will they'll do real-time corrections, but you're going to have to build those models off of existing data. Ideally, they would be built off your own data, because your behaviors won't be the same as other people's. So I think I have a personalized AI model around me, as you might imagine, that makes predictions. And these days, these things get pretty good. You can actually, get my company January Eye, and there's another paper out there now. You can predict people's glucose without a glucose monitor, believe it or not, from a smartwatch and other things. It's not as good as if you have a glucose monitor. Yeah, I think we can get quite predictive. Now, some things might be tougher, but a lot of the stuff should be.
Hi, thank you for a nice presentation. Actually, I have a question about the spikes you had with the age, about the aging. As my parents are getting close to the 60 point,
Yep.
I would be curious if there should be like 3 main points that are getting like bad in that age, so we know what to look for.
Yeah, I believe exercise is number one, and I think strength training is very important. So even though as a non-responder to lifting weights, I still lift weights every day, because keeping muscle mass up is really important to avoid sarcopenia. We knew that even before my study, but I think it only emphasizes that. And there's so many, there's now, including studies we're part of, that show all these, they're called exerkines, all these beneficial molecules like mitokines, things that are made by your muscle when you exercise. Also, your blood flows better, and keeping your microvascular system up is a big deal. So I think exercise is the most important thing. And even when people get old, you know, people often say, well, Grandma, it's okay, take it easy, sit in the chair. No, that's actually bad advice. Grandma, get up off that chair, start moving around the room, because people are active, just keep going and going. So, and you'll see it, right? People will live, people are always happy to live as long as they're healthy. And I think once their health plummets, then their desire can go down. So I think that's the key, keep people healthy. So that was one. So you want 2 more? Well, I'm a big believer in nutrition and avoiding ultra-processed foods. No, I don't know how much you can reverse that when you're 70 or something, I'm not sure. But probably, so that's good. Yeah, the 4 I said are a big deal, and especially, again, this whole community stuff that was described earlier is a big, big deal. So people, as soon as they isolate, you know, if their partner goes away, and they actually, their health declines. So it's really important to keep your social network up. Yeah.
Thank you.
It's kind of tough for me. I wasn't that social as a kid, but a little more so now, I suppose.
First off, I would like to thank you for the lecture. I just wanted to ask you, with all the biomarkers you're checking, is it possible that there are some spurious correlations as well, that, you know, when 2 variables are correlated, or at least they seem correlated, but in reality,
yeah. Well, I'm sure that's true. Again, we try to make sure our results are statistically significant, but whether some individual molecule, you know, if you have after a correction of p 0.05, some things that sneak in there will be by chance. If you're going to turn this into a clinical test, you're probably going to want to validate them on a second cohort and such. And a lot of these things, they are getting validated now. There's getting to be a lot more of these monitoring studies out there. So I think we will be in a world where it's going to be easy to do the validation. So, but you do have to watch. Yeah, we're literally making billions of measurements, and we use about 135,000 for the aging, actually. So,
thank you.
Sure.
We're just curious, looking at your data. I mean, rates of some cancers are rising. For example, the pancreatic cancer. Any clue from your data that what could be the cause?
Sorry about that. What about the cancer?
The pancreatic, or some cancers are rising without explanation why. So what comes to my mind is the pancreatic cancer.
I missed part. Some cancers are what?
Rising.
Oh, rising.
Yeah.
Oh, yeah. I think colon cancer is one of the biggest ones on the rise. So I think we eat terribly. I think the Western world eats way too much sugar, not nearly enough fiber. Fiber is huge, right? Fiber is feeding your microbiome, and there's some studies showing if you're not feeding your microbiome is eating at your intestine. And also, you know, and we have a study on this, fiber promotes a lot of these short-chain fatty acid producing bacteria, which are great for your immune system, and we showed for intestinal health. We even have direct mechanisms. They make short-chain fatty acids will show up on histones that are on these, you know, genes involved in intestinal integrity, like gap junctions and things like that. So I think fiber is a no-brainer. And again, I don't know about Czech, but in the US, it's recommended women get 25 grams, men 35 a day, and the average person gets about 12. So either about half or less than half, depending whether you're male or female. So we're just way off on our numbers. And it's been estimated that the diversity, the microbiome diversity of, Western people is about a third of what it was in ancient times, where people, these native cultures have a very diverse microbiome. Western cultures, it's not so good. So we're going to have to figure out how to repopulate, but to be honest, you're going to have to repopulate. Your microbiome gets set in the first 3 years of life. So we're going to have to do it in the next generation, I believe, to get full recovery. Right now, yeah, I'm not saying it's too late for all this. That sounds too morbid, but yeah, I think you should still take care of your health. But if we want to get all the way back to early days, you might need a better microbiome.
Good evening. Also, thank you for a wonderful presentation. I was wondering, you had some big cohorts, many participants, and do you still find it hard to gain all these people into your studies, or at this point the people are coming to you to know something about themselves?
Yeah, well, some are dropping off now. The original 109, when the pandemic hit, some never came back. We weren't allowed to keep sampling, unfortunately. Of course, it would have been the best time to be sampling, but we weren't allowed to. So, so we did lose some then. We've added on some new ones. Some of the tricks we do, because people learn health information, they get some information back right away. We're trying other things like to give them, even though I said the microbiome's not so easy, we have generated some microbiome reports to give back because people find it amusing. And so we do a few tricks. But I think the key for us, and be interesting to see what you find, is we hold annual meetings of the participants. Say, look, thanks to you guys, here's what we've learned from you contributing samples. And we've gotten some nice findings from this that we publish, and then we relay that back to people even before we publish it to say, look, thanks to you, we can learn a lot of pretty cool stuff. And they really like that. So those are some of the tricks you can do, but we still lose people. So far, only one person's dropped out. You're allowed to, in the US, drop out and ask that your data be no longer used. Now, it's in the consent. If they've already made it public, that's impossible. But yeah, only one person has done that in the whole time we've been there. Some people have just stopped. They said, it's a lot of work to do what I just said, to come in and give blood all the time. Yeah. So.
Thank you.
Yeah.
Good evening. As a medical student, I would like to ask you what skills are valuable for future physicians. What should I be practicing more or like trying to learn when I want to be a good physician in the future you are proposing?
Oh, you want to be a good physician? Take statistics, and I used to say learn how to code and work with data. I'm not so sure that's so critical anymore. I think you could probably do this vibe coding like this class I was referring to and get by with that. But I think you do if there's still some advantages, at least for the moment, coding. I think the key, again, I can't speak for here, but I can say for the US, it's the whole attitude's wrong. It's this whole, like they say, do no harm. That's why they didn't like sequencing genomes, is why they don't like whole body MRIs. All these things are harmful. And I would argue not doing things is harmful. So I think having the attitude of new is okay, recognize the limitations of that, make sure you convey that to patients and things. But I think being open-minded, yeah, I mean, I've had physicians scream at me why we shouldn't be doing genome sequencing. I don't think that happens anymore. And yeah, so I think keep an open mind. Yeah, the number one thing you could do. I'll tell you one other interesting story if you want to hear. Eric Topol wrote a book called Deep Medicine, about bringing AI into medicine. And he said, AI will replace everything except for empathy. But papers have now come out saying AI is way better at empathy than physicians because they're listening, they're responding the way the person can listen to. So, yeah. So keep an open mind.
Is it still okay, the questions? I mean, the amount.
Yeah, I imagine we're right. I mean, you guys are very kind to hang around, especially in a warm room, but sure.
Okay, so maybe the last one.
Yeah, but again, I'm not offended if people want to leave, yeah.
Okay.
Sure.
Hi. What has been your approach for genetic differences in different racial groups? Because
my approach for
your research, because when I've come across to some GWAS for different phenotypes, I've seen that a lot of the GWAS have been done with European descendant groups, and some of them, if they have been done with other racial groups, they have found differences. And I wanted to know what your approach with that has been. Like when you do your research, have you found any differences?
You mean, how do I decide what problems to work on, or how do I?
No, for example, when you think about, for example, the AI that does some predictions, I'm guessing these predictions are going to be different depending on your race, or even if, for example, if somebody's mixed race, what's going to be like their predictions? Have you thought about that in your research or in the wearables that you were thinking?
Yeah, well, the thing is, so personalized predictions, for example, what you would do is we'll all have some sort of predictive models about ourselves, like an AI agent, as I say. And it's very easy, whether you try or not, to perturb those predictions, right? Because nobody behaves perfectly. So you can just, like when I travel, I don't eat as good as when I'm at home, and I can see what shifts. So I think you will be doing perturbation experiments on yourself, both willingly and unwillingly. If you have problems, you'll probably experiment. Most of us do, right? If you have, for example, digestive problems, you will change what you eat, or if you start taking medications that cause problems. People experiment with that sort of thing. So, and I think if we can collect that information, you can learn better from it. If you don't collect it, you're kind of guessing, and then that's not so good. So anyway, that's one example of how you test your models. I think at the personal level. And the other way, of course, is to find people with similar patterns as you and see if they give similar responses. Yeah. It's complicated because there are a lot of variables that separate you from, again, the person sitting next to you.
Okay. I think that this discussion could be never ending because this topic is really interesting, and it's amazing that Professor Snyder is here, but I think that it's time to finish this lecture. So thank you for coming. I'm really happy that the young doctors who will treat us in future are here, and they start to think in a new way. And thank you, Professor Snyder, that you came.
Thank you for having me.
Thank you very much.